Hello, everyone. My name is Nuala Oleary, and I'm product owner for NCBI Datasets. It's a new and developing resource for getting genome data from NCBI. I'd like to thank the organizers of the Biodiversity Conference for giving me this opportunity to share the work Datasets has been doing on providing easier programmatic access to NCBI data. I'd like to start first by explaining what Datasets resource is. It's a relatively new resource that allows users to explore and download NCBI sequence and metadata in an easier and more intuitive way. We're achieving this through several approaches. We're developing new interfaces for accessing data. We're developing new mechanisms of data delivery that support delivering data at scale. And lastly, we are providing new file formats for metadata that are more versatile, along with better tools for working with metadata. The resource was first introduced in 2020, in March of 2020, and it continues to be under active development. We currently offer datasets for genomes, whole assembled genomes, for genes and orthologs, for organisms across the tree of life. We're also working closely with our users as we develop. So if you have any suggestions or comments, please reach out to us. We would love to hear from you. So there are three mechanisms for getting data from datasets. You can use our newly designed web interfaces. You can use our command line tools, or you can use our open documented API. The API serves as a single access mechanism for all datasets-based applications. Therefore, regardless of which interface you use, you're going to get the same data. This talk will focus primarily on our command line tool. We have two tools, one data format for getting data, and the other data sets for getting data, and data format for working with metadata. Before I begin to talk about the command line tool, though, I will first want to review what type of data you can get through datasets and how it's delivered. Datasets delivers data as a data package, which are returned as zip archives containing both data and metadata files. To date, we have three distinct packages. These are for genome, for assembled genomes, for genes, including orthologs. And then we also have a separate virus package, which we designed to support easy access to the SARS-CoV-2 genomes. Each interface allows you to pick and choose the file types you want in each package, along with the metadata. The metadata files are returned in JSON or JSON lines format, and these metadata files consolidate lots of information that was historically stored across different files on FTP or in different NCBI databases. And just to look more closely at what I mean by a genome data package. So for a single request, for example, the human genome, you can get a data package that includes a diverse set of files. This can be genomic FASTA or transcript or protein FASTA. It can also have annotation files. And again, it also has that metadata. You can request a package for a single genome, or you can request a package for tens or hundreds of genomes. This diagram here shows the structure of our command line tool and how it's organized. We have two top-level commands, download for getting data and summary for browsing metadata. Each datasets package can be accessed through subcommands for genome, gene, ortholog, and virus. You can query each package by for common terms using terms like assembly accession or bioproject accession, taxon for genomes, or gene IDs and gene symbol for genes. Each subcommand has multiple flags that can help you narrow down the desired genomes or genes of interest and the file types you want to include. You can also use the help flag at any subcommand to learn all about all the features that are available. The Datasets command line tool works with Mac or Linux or Windows systems, and our command line tool is also available as a conda package. So in the next few slides, I'm going to demonstrate how to use this command line tool to get a single genome or many genomes. So in this example, we're going to download the reference genome for the snow leopard. So the first thing we're going to do is use that summary command to find out if there is a genome submitted for snow leopard. So we'll use dataset summary genome taxon, and we'll put in the taxon name for snow leopard, and we'll limit it to the reference. Once we find out that there's the information, there is a genome for snow leopard, we'll then use the download command. And again, it's datasets download genome taxon with the taxon name, and we'll limit it to reference, and we'll name the file name snow leopard. And then we'll unzip the file. And you can see here in this example that all the files that you requested are in this zip archive. But perhaps you want to download not just one genome, but you want to download a genome for all reference genomes for Felidae. So you can again use our datasets download command. And this time for the taxon, we're going to put in Felidae. In this case, though, we're going to use a dehydrated command. The dehydrated command, this does is it gives you a zip archive that contains the metadata reports and then a fetch file for retrieving the data. We'll then use a rehydrate command. The rehydrate command will take that fetch file and will realize all the data files. The advantage of using the rehydrate command is that it has a lot of built-in retries, and it also downloads the data in parallel. So the data download is not only more reliable, but it's also faster. In this slide, I'm showing you an example of using the data format tool. Datasets, this is a companion tool to datasets, and allows you to export metadata from the JSON and JSON lines files and into tabular format, either TSV or Excel. You can extract information from the data reports or just all the information from the data reports, or you can select the relevant fields that you need. In this example, I'm using the JSON lines file that we downloaded for all Felidae, and I'm extracting a few fields of metadata and displaying them in a tabular format. To learn more about datasets, please visit our documentation pages where you'll find information on installing the command line tool and also how-tos for carrying out common tasks and a complete reference for all the command line subcommands and the flags, as well as our metadata schemas. We also have a GitHub repo where you'll find a folder containing training materials. These include Jupyter notebooks that will help guide you through some of our command line tool features. In the last year, Datasets is also now available in Galaxy platform. You can access datasets directly in Galaxy. Therefore, any workflows that require the input of NCBI genome data can now be run entirely in the Galaxy environment. And lastly, I'd also like to mention that Datasets is contributing to a new and exciting initiative at NCBI called the NIH Comparative Genomics Resource, also known as CGR. This is a multi-year National Library of Medicine project that aims to maximize the impact of eukaryotic research organisms and their genomic resources for biomedical research. NCBI is charged with leading the development and engaging the genomics communities. And if you want to know more about this resource, please visit the CGR website, and also please attend Valerie Snyder's talk at this conference and Sanjita Rangwala's poster. They will be describing the CGR project and some new development in our visualization tools. And lastly, I'd like to thank my team. And again, please contact us if you have any questions about datasets. We're always willing to listen to our users and find out new things that they need. So thank you very much. I'll take any questions.